Have you ever really stopped to think about that invisible world, the one thriving right inside you? I mean, take your gut, for instance. Trillions of tiny residents there. But what if?
Oh.
What if some of those microbes were secretly kind of driving one of the most serious health problems globally without us even having a clue?
Welcome to the Deep Dive. Today, we're really plunging into some fascinating new research. A study that seems to have uncovered exactly that a hidden culprit behind diabetic kidney disease. And this isn't just about, managing symptoms, it's about potentially finding a fundamental cause. A direct line from our gut bacteria to our kidneys. We're drawing on work published in Nature Communications. It's a collaboration between scientists over at the University of Illinois Urbana-Champaign and Me University in Japan. So our mission today, we want to unpack how this specific molecule made by gut bacteria seems to travel to the kidneys, how it causes damage there, and really importantly, how this whole discovery might be opening up completely new ways to treat it. Maybe even, a solution we've been missing for a long time.
It's such a critical area because diabetic kidney disease, and specifically the fibrosis, the scarring part, it's a really serial complication of diabetes. And it's, well, alarmingly, it's one of the top causes of kidney failure worldwide. The impact is just huge.
That sounds,
Yeah.
incredibly serious. It really hammers home why finding something new is so urgent, doesn't it? Beyond just keeping blood sugar in check. Because the alternative, facing dialysis or needing a transplant, that's completely life-changing for people.
Absolutely. And the tough part is a lot of people who've had diabetes for a long time, they eventually develop this.
Oh.
And once that scarring progresses, the options become really, really limited.
Yeah.
Like you said, it often leads to dialysis or needing a kidney transplant just to survive.
So with the treatments we have now, where are the biggest gaps, the real challenges?
Yeah.
Because it seems like we're doing okay managing blood sugar, blood pressure, which are obviously important, but maybe not getting to the core of the kidney damage itself.
Exactly. That's the crux of the issue. Dr. Taro Yasuma, he's a physician, and one of the study's first authors, he put it very clearly. He said, basically, there's no cure that stops or reverses the scarring or fibrotic process. And that's been a massive hurdle for medicine because the actual factors, the things driving the damage deep inside the kidney, they've just been, well, kind of murky until now. We've been managing the environment, you could say, but not stopping the specific molecular hitman doing the damage.
Okay, so given that huge challenge, this new study seems to step right into that gap, and it introduces this potential game changer, a bacterial molecule. Corisin, you said it was called.
Octaway, Corisin.
And it's not some complicated lab chemical, right? You mentioned it's peptide, like a small protein piece.
Precisely. A small peptide. And get this, it's generated by Staphylococcus bacteria. The ones just living in our gut normally.
Staphylococcus, isn't that the one we hear about with, like, skin infections? Or is this different?
That's a good question. It's the same broad family, the genus Staphylococcus, but the study's looking at species that are typically just part of our normal gut microbiome. Usually they're harmless residents there. But this research shows how even these normal gut bugs can produce stuff that causes real problems elsewhere, especially under certain conditions like diabetes. And, the idea that Corisin might be involved wasn't totally out of the blue. There was earlier work by researchers like Isaac Can at Illinois, showing Corisin could damage cells and make scarring worse in other parts of the body. So they had this suspicion, this hypothesis. Maybe it's doing the same thing in the kidneys. Maybe it's this hidden driver of kidney fibrosis they'd been looking for.
Ah, so they had clues pointing in this direction already.
Right. It was a logical next step, and it really seems to have paid off. To really nail it down, the team did some pretty rigorous work. They looked at blood and urine samples from patients who already had diabetic kidney disease. And the findings were quite striking, actually. They found much higher levels of Corisin in these patients compared to healthy people. And here's the key part. The amount of Corisin in the blood directly correlated with how severe the kidney damage was.
Wow, a direct link.
A direct link. And they saw very similar things happening in mice that had kidney fibrosis too. So it wasn't just a coincidence. It was looking like a real player in the disease.
Okay, the evidence sounds strong. Corisin is there, and levels match the damage. But the big question for me, and probably for you listening, is how? How does something made by bacteria in your gut actually get to your kidneys and start wrecking things? That journey seems incredible.
Yeah, that's the really fascinating mechanism they uncovered. It involved a lot of collaboration. People like Diwakar Shukla's group at U of I, using complex computer simulations combined with careful lab experiments, really tracked its path.
What was the trickiest part? Figuring out how it travels.
That was a big one. Small peptides like Corisin don't usually just float freely in the blood in large amounts. They often need a ride. And they found out that Corisin is pretty clever, actually. It latches onto albumin. Albumin is one of the most abundant proteins just circulating in our blood all the time.
Oh, right, albumin. So Corisin just grabs onto it.
Essentially, yes.
Hmm.
It hitches a ride, like it's catching the albumin bus through the bloodstream, gets access everywhere blood flows, kind of hidden.
A biological Trojan horse, almost?
Yeah.
Sneaky?
You could put it that way.
Okay, so it hitches a ride on albumin, travels through the blood. What happens when this package deal, the Corisin albumin complex, reaches the kidneys? Does it just dump its passenger?
That seems to be exactly what happens. When it gets into the kidney's filtration system, something about that environment causes the Corisin to detach from the albumin. And then this is the critical step the freed Corisin gets inside the delicate kidney cells.
And that's when the damage starts.
That's where the real trouble begins. Corisin seems to accelerate the aging process in these kidney cells. It pushes them towards senescence, basically. These aged senescent cells stop working properly, and worse, they can start sending out inflammatory signals, calling in more trouble.
Inflammation and then scarring.
Exactly. That inflammation leads to the buildup of scar tissue, that's a fibrosis we talked about, and as more scar tissue builds up, the kidney loses its ability to function properly. So it's this whole destructive chain reaction, starting with Corisin getting into the cells. It directly causes cell aging and death, kicks off inflammation, leads to scarring, and ultimately kidney failure. They really mapped out the how, turned that initial suspicion into a proven pathway of damage.
Wow. Okay, that pathway is clear and frankly pretty devastating. But the good news, I hope, is that the researchers didn't just stop at identifying the problem. They looked for solutions, too.
They did. And this is where it gets really hopeful. To prove Corisin was the main driver, and to test a potential therapy, they developed specific antibodies. Think of these antibodies as targeted missiles, designed specifically to find and neutralize Corisin in the body.
Okay, so antibodies designed to take out the Corisin. What did they look for in the mice that got this treatment? Did it work?
It worked remarkably well, actually. The results were pretty dramatic. The mice treated with these anti-Corisin antibodies showed a really significant slowing down of kidney damage progression. Dr. Esteban Gabatza from Me University noted that the antibodies, quote, slowed the aging of kidney cells and greatly reduced kidney scarring.
Reduced scarring, that's huge.
It is. They saw less inflammation, fewer of those aged senescent cells, and just overall the kidney structure looked much healthier compared to the untreated mice. It was strong evidence that if you can block Corisin, you can significantly interfere with the disease process right at its source.
That's incredibly promising from the mouse studies, a potential way to directly stop the damage. But, the big question always is, what about people? What are the next steps to see if this could actually become a treatment for humans? How far off might that be?
Right, that's the crucial translation step. And it's important to be realistic here. This is still early-stage research. As the paper points out, no such antibody is currently approved for use in humans. But, crucially, they also state these findings suggest it could be developed into a new treatment. So there's a concrete, hopeful path forward. It's not just theoretical anymore.
So what happens now? More animal studies?
Exactly. The immediate plan is to test these anti-Corisin treatments in larger animal models, like pigs. Their kidney systems are much closer to ours than mice. That's essential for figuring out safety, correct dosages, potential side effects, and just generally how it might work in a system more like a human's.
Makes sense. Laying the groundwork for potential human trials down the line.
Precisely. And it's also significant that the universities involved, U of I and Me University, have already filed a joint invention disclosure for these antibodies. That shows they really believe in the potential here and are taking the necessary steps to protect the discovery for future development. It's a serious move towards clinical application.
Okay, so let's bring this back home. What does all this really mean for you listening right now, and just for how we think about health in general? Clearly, it's a potential game changer for diabetic kidney disease, but are there wider takeaways here?
I think the biggest takeaway is this profound confirmation. Our gut microbes, the bacteria living inside us, have direct, sometimes damaging connections to vital organs far away, like the kidneys. It's not just about digestion. This fundamentally shifts how we should view the microbiome's role in overall health and disease. It suggests that maybe, someday, managing gut health could be a key strategy for preventing kidney disease. Think about targeted probiotics or even dietary changes, specifically aimed at managing these Corisin-producing bacteria.
That's a radical shift, moving from just reacting to kidney damage to potentially preventing it by looking at the gut.
It opens that door, yes. It challenges us to see the body as this incredibly interconnected ecosystem.
And the impact for patients, if this antibody approach pans out.
Oh, the potential real-world impact is enormous. If we could actually develop a treatment that slows down or prevents kidney scarring in diabetes, that would be revolutionary. It could vastly enhance the quality of life for patients who right now have very limited options once the scarring starts. It's about moving past just symptom management, like controlling blood sugar, to actually intervening in the core mechanism of the damage, addressing a root cause.
A targeted therapy like that could genuinely change millions of lives.
Absolutely. And then broader, too. This kind of discovery might lead to new screening tools. Maybe a simple blood test for Corisin levels could identify people at high risk long before serious damage occurs. That allows for early intervention, shifting the whole paradigm from reactive treatment to proactive prevention, all based on understanding these tiny molecular signals from our own gut microbes.
What an incredible journey today, from the hidden world inside our gut to the front lines of fighting a major disease. Unmasking Corisin as this gut toxin driving diabetic kidney damage, and then seeing the potential of these targeted antibodies. It's truly cutting-edge stuff, gives you real hope. So as we wrap up, here's something to think about. Imagine that immense power packed within your own body, those trillions of gut microbes. We're just starting to understand how deeply their activities, their conversations, influence our health everywhere, not just in the gut. Corisin is one example. What other hidden signals, what other culprits or even helpers might be operating silently within us, just waiting for science to uncover them? The answers could unlock entirely new ways to stay healthy and fight disease. Really makes you think, doesn't it?